whole area out there. That's your sclera. White connective tissue that maintains the shape of the eyeball. Right? That's why this whole sclera part is kind of white, opaque kind of color in there. Provides a site for muscle attachment to all those 6 strapped muscles we looked at, right? Provides a site for it in the sclera. Cornea, this is anterior 1/5 of the eye. Cornea that you can see from the front. Transparent and refracts light that enters the eye. So this is the part that's transparent, the layer. If you take it out, then you can see it's transparent. But this is where it refracts the light. The reason it's transparent and not white, like sclera, is because they say it has less water in there. So the reflection is, they're not reflecting all of the light. So when they do that, then you see it as white color in all colors. So when it's keeping some, then you don't see that as white anymore. So that's why it happens to be transparent here. Choroid, this is your vascular layer. So we have choroid, ciliary body here. Choroid, a vascular network. Many melanin-containing pigment cells are in your choroid. So this is what appears black in color, right? So this choroid is all over inside the second layer also that is black in color. Prevents the reflection of light inside the eye. Ciliary body, you have the ciliary ring and ciliary processes here. The ring is a thickened ring of tissue surrounding the lens, and it's composed of smooth muscle bundles, also called ciliary muscles. We'll look at a picture of that. Anchors the suspensory ligament that holds the lens in place, and it changes the shape of the lens. Like you need to change the shape of the lens for you to to see things like far and near. When you're looking at things, then your lens, it changes the shape. It can become like convex or concave, go in and out according to what you're looking at. So that helps with your ciliary ring here. Ciliary process produces aqueous humor in your eyes. Iris is also another vascular layer. This is the smooth muscle regulated by the autonomic nervous system. You have sphincter papillae and dilator papillae in your iris. Sphincter papillae is when the pupils constrict. That's your sphincter papillae doing that. Closed vision and bright light, right? Like if you flash bright light, you see your iris constrict, right? Or closed vision constrict. When it's distant vision or dim light in general, your pupils dilate, stay dilated, and that is done by your dilator pupillae. Changes in emotional state. Also, pupils dilate when the subject matter is appealing or requires problem-solving skill. They obviously did research on that to know that. So also changes in emotional state causes changes in your pupils dilating. Controls the amount of light entering the pupil. That is one of the major functions there. It's like the camera. How you have the shutter space. You can, like when you close it, you can see it closing, and you can control. And also microscope has it, right? How you control the light coming in and out. The iris diaphragm, we call it. So this is how your iris is controlling the light entering and how much of it is in there. Color is determined by the amount of melanin present. That's what gives you your eye color that you see. Some of us have brown eyes, some has lighter eyes. It's the melanin. So when you have a lot of melanin in your eye, then you get a darker appearance, a brown color. When you get less and less, then it becomes different colors. You have blue and this green. And if you don't have melanin at all, a condition when the person is albino, no pigments at all, then they actually get this eye color that's kind of pinkish. it's showing your vascular color. I guess it's pinkish. That would be interesting to see. I always thought. Anyway. So here on this picture, you're looking here at the pupils and the sphincter papillae, the muscles that constrict it, and dilator pupillae that helps it to dilate. So those are the muscles in your pupils. This is where we're looking at the ciliary ring and the ciliary processes, and the lens here, how it's holding it with the suspensory ligaments. So the lens can change shape, convex or concave. Here you're looking at this vitreous chamber, so this way it's cut in the sagittal side again. So you're looking at vitreous chamber and the posterior and anterior chamber in there. The ciliary body here, it's ciliary ring and the processes down here. You have your retina inside and the choroid, that's the black colored area. The nervous layer now. So we looked at your vascular layer, we looked at fibrous layer, right? We're looking at the nervous layer. This is your retina, right? That holds all of your cells that we talk about in your nervous system. The nerve cells are your rods and cones. The inner layer of the eyeball is your retina. So the very inside layer has over 126 million photoreceptor cells which respond to light. Wow. 126 million photoreceptor cells. It's a lot. Macula, or the space fovea centralis, is the area of greatest sensitivity to light. And we'll look at a picture of that. But when you look at your retina, whole space, the whole ball area of your area of the retina, there's one small space at the back where you have lots of those photoreceptor cells packed in there. One little tiny area at the back, which we call fovea centralis. This is the area of greatest sensitivity to light. Highest concentration of photoreceptor cells. Then you have the optic disc close to your macula and the other side where you have the optic nerve exiting from. This is where location through which nerves exit and blood vessels enter the eye, the space there, nerves and blood vessels coming in and out. No photoreceptor cells in that area at all. And this is what we call the blind spot of the eye. Did you try that test ever to find your blind spot? You should do that. All you do is, if you have a white paper, you can draw a black circle in one side and maybe a cross on the other side. Close one of your eye and bring this paper close, around to find your blind spot. Focus on the cross. You'll see in one of the positions the dot you make, the black spot, will disappear. You have to find it, like you hold it and you look around till you find your blind spot, and you won't see it. You can hold it there, you will see nothing there, it will disappear. It takes a while, but that's your homework. So try that out tonight. It takes a while for me. Every time I'm like trying to find my blind spot and then my eye gets tired. But that's the spot when where you don't see anything, and the reason is that's where your optic nerve stuff is coming in and out from. So that's like a space hole where you don't have any of your photoreceptor cells, right? So how are you going to see without something receiving it, right? So you can't see it in that spot at all. So this is the picture, and this is your blind spot test. So you look at this cross here, and this red spot will disappear in a position when you close one of your eyes. So this is your macula, or the fovea centralis, the center part of the macula. This is where your photoreceptor cells are packed in. Back here, close to your fovea centralis, is the optic disc, the whitish lighter area that your optic nerve stuff goes through, and it's your blind spot basically. You don't see anything there. You can see all the vessels here, retinal vessels in your retina area as well. Chambers of the eye, composed of 3 chambers. So you have the anterior, posterior, and the vitreous. chamber. Anterior chamber between the cornea and the iris, the front one.